теперь мы переходим к научной части нашей конференции. Первую пленарную лекцию делает профессор O'Brien. The taming of the cat.
I will begin by thanking Professor Kolchanov and Tikhonov and Borodin and Rapidotsky and the organizers of this conference for the honor of being able to talk to you at a conference honoring Dmitry Belyaev, the classic domestication of the silver fox, which is a canid, and you're actually asking me to talk about cats, which is a big surprise. I'm delighted to accept because Professor Belyaev and the influence of the Novosibirsk domestication genetics community stimulated many of the ideas that I will present. And basically, the major idea is an observation that domestication of plants and animals, the natural domestication, which began about 10,000 years ago in the Middle East, was perhaps one of the most important inducers of human civilization. And it was really an experiment in genetics by the first geneticists, the hunter-gatherers who settled down to build village in the Middle East and to take the wild animals that was their nature and to use them for human betterment and civilization. These animals included barnyard animals such as sheep and goats, and cows, horses, and camels, and many, many other species. But one animal that was domesticated before the intentional artificial selection in the Middle East was the wolf, because young wolves were trained and adopted, and they had a function. They guarded the settlements, they helped hunt, and they gave companionship to the early hunter-gatherers. This was maybe 5,000 years before people themselves were domesticated. They were wandering around in the world as hunter-gatherers. Today, there are maybe 100 different breeds of domestic dogs, all selected by artificial breeding for many, many characters. And 10 years ago, the full genome of the domestic dog was sequenced, and it gave the veterinary clinicians who have studied diseases in dogs and characters in dogs a terrific boost that allowed for this species to become a powerful medical model and a model for behavior and a model for other things. Dmitry Belyaev anticipated this before I was born, because he knew something was very interesting about domestication, and he wanted to know more. I'm going to tell you about cats, a different kind of domestication, not one that was driven by utility. In fact, most people will argue that cats have no utility whatsoever. They just wander around and look kind of pretty. In the 19th century, people started to breed them in cat shows. The first one was in the Crystal Palace in Britain in 1871. And since then, there's about 60 breeds of cats that wander around. But the cat also survives fine in the streets of Novosibirsk, in the streets of St. Petersburg, in the streets of Johannesburg and Rio de Janeiro, as feral, released wildcats, remembering their wild nature, which their predecessors had. If you look with molecular tools at the 60 breeds of cats, you can discern and develop certain characteristics that relate Oriental cats to each other and other breeds of cats that were developed in Europe, in the United States, even in Russia. But that's not the interesting question. The interesting question is, where did the cat, a wild animal, unusual for a domesticated species, come from? Most domesticated species are herd animals. They behave well because they get along with each other and they have a leader. And domestication of horses and cows and sheep was a matter of changing the leader from the lead cow to some person. Cats don't do that. They don't like leaders. They're isolated. They set up territories. And they're not a good candidate for domestication. And believe me, not all species can be domesticated. We can tame a tiger if we hand raise it, or a cheetah, or a chimpanzee. But that's not domestication, because they don't transmit that information to their offspring at all. And we learned in the 20th century, through the investigation of premier geneticists here and in the United States and other places, that domestication is about genetics, even though Mr. Lysenko might have disagreed. Now, most people know that there's a lot of species of cats, but many of them are unrelated to the house cats. But the parent cat of the domestic cat is thought to be a group of species that exist in South Africa, in Asia, and Central Asia, such as the sand cat, the jungle cat. You'll see these species in zoos, and they look a little bit like house cats. Now, most people agreed that the parent species of the house cat was the one that looked most like it, the European wildcat, which also exists in Asia and in Africa and in China and in South Africa. It has subspecies names indicated right here. And the relationship to these is something that our group became interested in molecular terms. By applying molecular genetic techniques to populations of 985 cat specimens that were collected across the range of the cats, we came to the conclusion, and I'll tell you the simple conclusion first, that we could tell the cats in Europe, purple Sylvestris, apart from the cats in China, the red ones, or the cats in Eastern and Western Asia, the Felis ornata, or the cats in the Middle East, Felis libica, or the cats in Southern Africa, Felis Sylvestris capra. Now, that's the result we're heading towards. Now, most anthropologists have been fascinated by cats for thousands of years. They've been in our art, in our literature. In Egypt, they deified and made gods out of cats. And most workers and experts agreed that the origins of cats was probably in Egypt, because it was in Egypt that they mummified cats, and they counted cats by the ton as they collected mummies and distributed them throughout the world and into the museums. And everybody thought it was settled. However, my student, Carlos Driscoll, he didn't believe any of it. He wanted to see himself. So he set out a graduate program that lasted him quite a while, where he traveled across the range with his colleagues and collected specimens from cats in the wild, cats in museums, and domestic cats. And then he used the tools of molecular evolution to come up with a structural distinction between the subspecies and relate them to house cats. He published this originally about 6 years ago in Science. There was a review article in Scientific American and a more definitive article in PNAS. Basically, he collected animals from Europe, from the Middle East, from Asia, from Southern Africa, and from areas throughout the range of the domestic cats, doing interesting things like scraping out things on their tail. He applied the tools that we knew could discriminate between populations, such as mitochondrial DNA, using 2,600 characters, of which almost 200 were informative. He also used nuclear loci, stutter repeats called microsatellites. These are the hypervariable DNA segments that are used in DNA forensics for personal identification in homicide cases. He used 36 of these that were mapped to many different chromosomes, and he used tools, algorithmic tools, to do the analysis to set up the population structure, including phylogenetic analyses and something called structure. Now, what he discovered, as I saw, was that there were signals in these cats. If the sand cat, which is an outgroup, was the basis, he found that he could tell the difference with specific markers of Chinese mountain cats, European mountain cats, Southern African cats, Central cats, and Middle Eastern cats. I might say that there has been 30 years of attempts to do this with dogs, and they have never seen a signal, because the evolution of dogs from wolves has been so deep in history that most of the signal went away. Add that to the development of the dog breeds and the signals that were unique. This is not true with the cats. The cats still retain their ancestral markers, and they allowed us to make this discovery. The next slide is very detailed. It consists of a phylogenetic or evolutionary tree of all the cats in Carlos's study. This is actually a tree based upon mitochondrial DNA. And the colors are important here. The outgroup is the sand cat, which estimated to have diverged from all the other cats over 200,000 years ago. Then a radiation began that led to the European wild cat, the South African cat, the Central Asian cat, the Ornata, and then the purple ones here are from the Middle East, but the brown ones are all house cats, inbred house cats, feral house cats from throughout the world, and none of them align with the Asian wildcat or the African wildcat or the European. They all align with the ones from the Central East. In fact, we couldn't tell them apart with molecular genetic data, as if the closest relative to modern domestic cats is something in the Middle East. So another point to make is, when Carlos began this study, he got a lot of pushback from his expert advisors who said, Carlos, this is a very stupid project. Let me tell you why. Domestic cats are living all over the world, and these feral cats and these wild cats that are supposed to be progenitors, they interbreed with them all the time in nature. So you've got admixture, gene flow between feral house cats from downtown London and the wild cats out in the countryside, or feral cats somewhere near Saudi Arabia and the and the feral cats in the cities of Saudi Arabia and the wild cats that live out in the desert, you'll never see a signal. He didn't believe it. And actually, this is a very interesting slide because it gives a color code to the fraction of individuals in this particular area, Spain, for example, that had the European specific signature. These are green. The brown is the domestic cat signature. So some of the cats had a ancestral European signature and others were domestic. So they were right. There was mixture going on. But Carlos could sort it out because the green individuals only occurred in green. The red individuals only occurred in China. The purple, which is Ornata, only in the Far East. The brown, Libica, well, they were in the Middle East, but they were also in the house cats, remember? And of course the blue ones were down in southern Africa. So this kind of study led him to think, there is a structure and the house cats are all closest to the ones in the Middle East. Well, that's pretty cool. The microsatellites, or short time and repeats, said the same thing. When we clustered individual animals, a composite genotype of 26 markers, what we found was that every European wildcat had as its closest relative another European, and every Southern African was closest to the Southern African. But the house cats were mixed in with the Asian ones from the Middle East, from the so-called Fertile Crescent, the same place where humankind began. When we use the structural analysis, which is a tool that geneticists used to color different individuals, we found the same thing with the microsatellites. The wild cats broke up into structures like the breeds, and the domestic cats were broke up into breeds, but they were different from the wild cats. Now, when we looked at the dates of origin of diversity in house cats and wild cats, it came out to be on the order of 150,000 years ago, which didn't make any sense because there was no archaeological evidence of any association of house cats with people until about 9,500 years ago. So the diversity we were seeing was probably the diversity that was present in the parent wildcat population that went back much further. That's a technical point, but it explains an uncertainty we have. When we actually looked at the archaeology, Jean-Vincent Vignet from Paris published an interesting finding from the island of Cyprus, where he saw a child that was buried, and next to it was a house cat, a baby house cat with some bracelets and other things in here. Jean-Denis Vignet published this in 2004, and it is the earliest and the oldest date of the association between human findings and cat findings, 9,500 years ago. The beginning and the end of the Egyptian era was 3,500 years ago, much, much later. So maybe it was wrong. What happened was the research uncovered the carefully buried cat on Cyprus. Cyprus is an island that doesn't have any wild cats. The only place this cat could have got there is on a boat with somebody. So by this time, the cats and the people were already talking, or at least going with each other on boats. The Egyptian period was much later. It was about 5,000 years later, and as I mentioned, the cats were venerated there, and they were made into gods, and there were strict laws against transport of cats. They didn't work very well because the cats made it into Rome, and then when the Roman civilization started to spread out, the cats went into Europe, they went into other places as well. So by and large, the 5 subspecies of cats were discriminated with the molecular markers. We were able to show that the domestic cats came from the Near East Asia. The archaeological records gave us a date and a place, the Middle East. And the 5 matrilineal events were much older, but that's because they dated with the origins of the cats that were there. So how did this actually happen? How did domestication happen? Well, I wasn't there. But this is what we think. And when I say we, I mean the community of people who have looked at all the data, the archaeological, the historic, and the genetic data. When humankind settled down from being hunter-gatherers wandering through forests, they would do something very unusual, which is they would build little teepees or huts, and they would live together in families, and then they would cook, and then they would harvest materials from the neighborhood, and even put up fences around some of the animals that were a little bit tamer. And they would basically develop ways of staying in the same place, settling down into agricultural villages. And in those agricultural villages, the number of domestic species became known. The wild einkorn wheat, the wild barley, the sheep, the pigs, the cattle. Domestication all took place in this region at about the same time. And it is exactly the same time as the oldest fossil of cats associated with human. So what we think is that the cats probably noticed these villages and wandered in because there was plenty to eat. But it was more than plenty to eat. There were other species that had settled down. This is what the first geneticists were envisioning someday, is to have organized agriculture to help feed the world. But settling down into the villages were the domestic mouse, Mus musculus domesticus. And these mouse certainly attracted cats, as did the trash tips on the outskirts of town. The cats wandered in, and some of them were different from the ones in the wild. The ones in the wild were kind of nasty and aggressive. The ones that wandered in did not eat the children. They made friends with the children, and they made friends with the people, and they became, over and over, selected at least slightly by the environment. So what happened is, in these environments, they would eat the mice, they would go to the trash, and eventually they were invited into the living rooms because they were very cute, just like the foxes. Okay? And eventually they would show up in the trash dumps. And there were a lot of funny things going on. But in these areas, when the trash was coming out of here, it represented an opportunity. And it was an opportunity that humans didn't intentionally do. Humans didn't intentionally breed cats. They had no windows, they had no locks on doors, and if any of you have ever tried to breed your cat without a window or a lock on the door, forget it. The cats will find their way to breed with whomever they like. And that's what these cats did. So, finally, one of the interesting things that came out of evolutionary theory in the 20th century was allopatric and sympatric speciation. The simplest idea of most species development was that they became population of one species would split by a river, and then it would evolve, separated over a long time into two different species. That was allopatric speciation. And there was some argument as to whether sympatric speciation could take place. Sympatric speciation means speciation without separation. That's what these cats did. The wild cats were out there, but they came into the villages and they evolved. We're not really a true species yet in the cats. It's only been 10,000 years. It takes 1 million or 2 to get a species. But this is a way sympatric speciation could have happened, by providing such a different ecological opportunity. And why didn't it occur over and over in Africa? Because the first villages, the first place that had the cats, that had the dumps and had the mice, that was in the Fertile Crescent. So it was easier to get on a boat and travel to Africa or to China than it was to evolve your own situation there. So, over the next few years, there were materials found in Israel up to 9,000 years ago, which again affirmed the origins in the Middle East, and in Egypt 3,600, Pakistan 4,000 years ago. America and Australia was very recently, within the last 1,000 years, but over Europe, the cats were distributed. In Egypt, It started 3,600 years ago, and then it became official deity in Egypt, and then began to be dispersed simply by the boats. So house cats were all over the place, and they began to be developed into breeds in the 19th century. So the cats that we see now are being selected for their appearance and for what they do. Cats are not the only species that became domesticated, but they're the only species that domesticated themselves. These are some of the other cat species that all had functions of some sort or other. The dog, the horse, the pigeons, the mice, the rats, goats, sheep, cattle, pigs. All of these have functions at feeding, clothing, work, labor, guarding, you name it, riding on horses. All of these are functions. Remember, the cat don't take instructions very well. So the cats didn't really have that ability. Now, so basically, recently, the genome sequences has empowered into the domestication questions. You'll hear more of this from Anna Karkova this afternoon and the group here. But we also were curious about the domestic cat, so we sequenced this guy, Cinnamon, who is a cat from St. Louis, Missouri, and published several papers improving the assembly and the annotation, including the search for genes that are interesting in the context of the domestication. When you publish a genome paper, what you do is you assemble it from these short reads into a long chromosome level sequence, and then you also identify all the genes and all the variants, and then you look for association that has either selected or has been somehow statistically unusual as indication of a recent event that may have favored it, such as selection. In the house cat, there's a dozen genes that have been identified that are associated with cat domestication behavior. Several of them have been implicated in human neurology, and we're not sure whether these are the ones, but we think because of the fact that they're long stretches of homozygosity, that they're good candidates that we need to do some more functional genetics on. And with the CRISPR technology available, I think it's going to be very shortly before we're going to see some of this happening. I think I'd like to now thank the many people who've helped us. Carlos was the leader of the domestication problem, Carlos Driscoll. He's now a staff member at the National Institutes of Health in the US. These are some of the gang that helped develop the cat map: Bill Murphy, Marilyn Raymond, Joe Meninger, Now Yuki, Eduardo Zurich, Warren Johnson, Jill Slattery. This is a group that I managed at the National Institutes of Health at a retreat about 10 years ago, and these are the people that work with me not only on human genetics but also on my interest and avocation on the cat family and on domestication. More recently, okay, boss. More recently, okay. Okay, I guess the rest of the slides aren't there. I was going to show you a picture of, just a second, let me see if I can find it. Yes, here it is. Yeah, this is a picture of Theodore Stubzhansky, the Russian geneticist who the institute I direct was founded in honor of. This is one of our workshops. These people also worked on the cat a little bit. And more recently, this same group has used the tools they learned from domestication to help start and coordinate a national project called Genome Russia to sequence about 3,000 Russian people, mine their genomes for medically important mutations, and to allow for the reconstruction of natural history, such as we just described for the cats, for the many different minor ethnic indigenous groups that live in Russia today. It's an exciting time for the Stubzhansky Center and for Russian genetics. Thank you very much.